Nearly 1 million Americans live with Parkinson's, and 60,000 new cases are diagnosed yearly. Chances of acquiring Parkinson's increase with age. Only 4% of the cases are diagnosed under the age of 50. Men are 1.5 times more likely to have Parkinson's than women. People of European or Hispanic descent are twice as likely of getting Parkinson's than either people of Asian or African descent. The majority of Parkinson's disease cases are non-randomly distributed in the Midwest and Northeastern United States. Approximately 15% of people with Parkinson's disease have a family history of this disorder. Researchers have identified more than 100 LRRK2 gene mutations in families with late-onset Parkinson's disease, the most common form of the disorder, which appears after the age of 50. Parkinson's patients with LRRK2 gene mutations are clinically indistinguishable from patients with Parkinson's of unknown origin. Scientists in Singapore developed a zebrafish model of Parkinson's disease by blocking the normal function of the LRRK2 gene. This resulted in Parkinson's-like locomotive effects and the loss of neurons similar to those of Parkinson's patients. Mitochondria are the human body's cellular power plants. Genetically modified fruit flies with a pink1 mutation cause mitochondrial defects resulting in a loss of their ability to fly. When the flies were given vitamin K2, the energy production in their mitochondria was restored, and the insect's ability to fly improved. Mutations in the parkin gene are the most common cause of juvenile and early-onset Parkinsonism. Mitochondrial dysfunction is a major problem for Parkinson's disease patients, and a loss of parkin function impairs the activity and integrity of mitochondria. Chromosome 6 contains the parkin gene, previously thought to be involved only in rare early-onset form of the disease, but the researchers discovered parkin mutations in 18 families, with both late and early onset cases, including 11 early onset and 7 late onset families. People who develop Parkinson's disease because of mutations in a gene called parkin better retain movement and cognitive abilities after years with the disease than did people with non-inherited Parkinson's. For example, the motor symptoms of people with parkin mutations are often not as severe and are better controlled over the long term with levodopa than are the symptoms of people with sporadic Parkinson's. Also, Lewy bodies, the clumps of protein that are a hallmark of sporadic Parkinson's, are rare in the brains of people with parkin mutations. In 2015, it was discovered a recurrent mutation in PARK2 is associated with familial lung cancer. The cohort study of Danish Parkinson's disease patients confirmed an inverse association with smoking-related cancers. This inverse association was first seen in 1966. Thus, there is an intriguing picture of fewer smokers among Parkinson's disease patients, and even fewer tobacco-related cancers among Parkinson's disease patients who smoked. In cancer, parkin was already a suspected tumor suppressor gene because it resides on the long arm of chromosome 6, a segment of which has long been known to be altered or deleted in a wide variety of human cancers. Chromosome 1 is a hotspot for Parkinson's-associated genes. The short arm of chromosome 1, signified by a P, position 35 to 36, is associated with PARK genes 6, 7, and 9. PARK 6 is associated with 4 members of a Sicilian family who were afflicted with early-onset Parkinson's. PARK 7 corresponds to Dutch ancestry and early-onset Parkinson's. PARK 9 corresponds to the north of Jordan and also early-onset Parkinson's. The short arm of chromosome 1, position 3-2, is associated with the park10 gene in 117 Icelandic patients with late-onset Parkinson's. The park9 gene in humans and a comparable gene in yeast cells, the YPK9 gene, protects against manganese poisoning in both humans and yeast cells. Both park9 and YPK9 are protective against alpha-synuclein misfolding, which leads to cell death. Manganese poisoning can induce Parkinsonism in miners and industrial workers by loss of neurons in the motor region of the brain, the globus pallidus. European Parkinsonism patients with the park9 mutation also lose neurons in this region. In people with park9 mutations and yeast cells deficient of the YPK9 gene, both experience cell death when exposed to manganese. How does park9 protect against alpha-synuclein toxicity? And how does park9 help prevent manganese poisoning? Allison Wright Willis, assistant professor of neurology at Washington University in St. Louis, found that high levels of urban pollution containing manganese and copper increase the risk of Parkinson's disease. Manganese pollution increases Parkinson's risk by 78%, and copper pollution increases Parkinson's risk by 11%. For approximately 60 years, lead was added to gasoline as an anti-knock agent until being phased out starting in the 1970s in favor of another anti-knock agent, manganese. In families with late-onset Parkinson's, the strongest overall evidence for linkage was on chromosome 17, near the tau gene, which has been implicated in other degenerative neurological diseases, such as Alzheimer's disease. Frontotemporal dementia and Parkinsonism is caused by mutations in the tau gene, and over 100 families with 38 different tau mutations have been identified worldwide. Frontotemporal dementia and Parkinsonism has poor or no response to L-DOPA therapy. A harmless human virus was genetically modified with a human gene that dampens down nerve cells, and was then injected into the brains of Parkinson's patients. The patients showed motor improvements from 30 to 65%, and brain scans showed some malfunctioning nerve circuits were working normally again. Macrophages are the largest white blood cells in the human body, and they eat pathogens and debris through a process called phagocytosis. Macrophages pass the blood-brain barrier and seek out inflammation. Macrophages loaded with the antioxidant catalase relieved MPTP inflammation in mice and resulted in a twofold reduction in the activation of resident macrophages, called microglia, and a twofold increase in the enzyme tyrosine hydroxylase. A bacteriophage is a virus that infects and replicates within bacteria. A modified bacteriophage, M13, significantly reduced alpha-synuclein, increased tyrosine hydroxylase, and improved motor performance in a preclinical model of Parkinson's disease following a direct-to-brain single injection. Austrian biotech company Epheris AG is developing a vaccine which introduces alpha-synuclein antibodies into patients with the intention of targeting and promoting the clearance of alpha-synuclein. The vaccine is proved to be safe in a phase 1 clinical trial. Jean-Martin Charcot is considered the father of neurology. From 1868 to 1881, his studies furthered the understanding of Parkinson's disease. Charcot led the effort to have paralysis agitans to be renamed Parkinson's disease after James Parkinson's. Charcot developed a vibration chair for patients with Parkinson's after he observed that patients were more comfortable and slept better after a train ride. He replicated the experience by making Parkinson's patients sit in the chair for 30 minutes a day. "40 Parkinson's patients were treated with a Physioacoustic chair, a chair that vibrates using low-frequency sound waves, and patients had a significant decrease in rigidity and tremor. Results of this initial investigation provide support for vibration therapy as a non-pharmacological treatment alternative.